Bjarni, your paper on the complete recombination map of the human genome is out now in Nature. And it's interesting because it is basically, if I understand it correctly, the first paper describing both crossover and non-crossover recombination maps of the human genome. What is the value of that?
So it basically tells us how people are an amalgamation of their grandparental DNA, and where there are changes between the DNA of your parents and your grandparents, and that helps us understand how human life is generated, and it helps us understand why it fails at some point.
And it's interesting because there are these two principal methods to generate new diversity in the human genome. It is the recombinations that create a mixture of haplotypes or sequence diversity from the chromosomes of the mother and the father. And then you have the de novo mutations. And the simple version of the story is that women generate diversity through recombinations and men with de novo mutations. But you tell us it is not quite that simple.
It's not quite that simple, no. So there are more of these recombinations generated by the woman, and there are more of the de novo mutations generated by the man. But interestingly, in this paper, one of the key observations that we found is that a large part of the de novo mutations are generated through this recombination process, and particularly in women, where, especially when they age, they start to generate a lot of de novo mutations.
And this is fascinating because we have been studying the generation of new diversity in the human genome for the past 25 years. And in some of our earlier papers, we demonstrate how recombinations are under genetic control, that there are variants in the sequence that affect both rate of mutations and location of mutations. And now you tell us that the recombinations themselves generate new diversity in the sequence, which is fascinating. So you have variants that generate recombinations, and you have the recombinations that generate variants.
Right. So, I mean, the DNA is somehow programmed to make itself vary, so we won't all turn out to be the same.
Yeah, and thank God. And it's more than that. If you look at the diversity in the rate of de novo mutations, almost 95% of that seems to be generated by the age of the parents at the conception of the children.
Right.
And then you also have the impact of the age of the parents on the rate of recombinations.
Yes, so recombinations increase with the age of the mother. And we knew this for crossovers, that there was a slight increase. But when we start looking at non-crossovers, which are these very short interchanges between the grandparental DNA, we see that there's a very rapid rise in those with the age of the mother. And this is probably because the mother has to keep the egg in a very safe in an environment for many years, and many more years if she is an older mother than if she is a younger mother.
So one of the things that is important to keep in mind, that the healthcare consequences, the healthcare, let's call it the cover charge that we pay for the ability of our species to generate new diversity in the sequence, which is necessary for continuing evolution and adjustment to new environment. The price we pay for that are the devastating syndrome of early childhood. It is spontaneous abortions. And more than that, it is even that some of these de novo mutations that have significant healthcare consequences are mutations that lead to the onset of lead to diseases of late onset. So they can continue to infiltrate the population and lead to a significant healthcare burden, public health burden in our society.
Exactly.
So, Bjarni Halldorsson, I think that this is a wonderful new addition to our very large number of papers we've been publishing over the past 25 years, trying to figure out how man continues to adjust to his environment through new sequence in the genome. So thank you.